Okay, I think it's about time that we get started. I'm Lynn Birnbaum, and it's really my pleasure. I cannot believe we are having an in-person meeting. This is so exciting. Whoops, let me take off my mask. Everybody, you must wear masks in the building except when you're speaking. So just please be careful. I mean, none of us want to get shut down again in any way, shape, or form. So I really want to say, when we set the date for this meeting well over a year ago, none of us believed, or none of us thought we'd still be in a situation that we are today. So it's very exciting that we are being able to go on. And I also want to thank all the people who are in the ether, and watching and participating virtually. And I will urge all of you to make sure, if you're virtual, that you put your comments right in the chat, you ask your questions in the chat, and folks from the National Academies will be collecting those and presenting them so that we know what your thinking are. So again, I really want to thank all of you for attending the workshop on the role of companion animals as sentinels for predicting environmental exposures, effects on aging, and cancer susceptibility in humans. Next, please. Next. Or do you want me to do it? I have the clicker? Okay, I will do it. So I really want to thank the sponsors of the workshop. It would be possible without the generous support of folks, and especially the work of Danielle Carlin of NIEHS and Rod Page of Colorado State University. So the workshop is really being hosted by the Cancer Policy Forum in collaboration with the Forum on Aging, Disability, and Independence, and the Standing Committee on the Use of Emerging Science for Environmental Health Decisions. Okay. So the National Cancer Policy Forum provides a continual focus on cancer within the academies. The forum is a trusted venue for experts to convene and identify high-priority issues in cancer research and care. The forum then works collaboratively to examine these issues and identify potential opportunities for action. And again, the work of the forum would not be possible without the support of their sponsors, very generous sponsors who are listed here. Now, the Forum on Aging, Disability, and Independence provides a natural venue to discuss issues that affect an individual's ability to live independently, focusing both on individuals who are aging with a disability and individuals who develop disabilities as they age. The work of the forum, again, would not be possible without the support of their sponsors listed here. Now, the role of the Standing Committee on the Use of Emerging Science for Environmental Health Decisions, I guess I'll have to say this is kind of an NIEHS-funded effort that has been going on for many years, examines and discusses issues on the use of new science, tools, and research methodologies for environmental health decisions. Now, the Standing Committee for the Environmental Health Matters initiatives is something that actually I played a role in starting to initiate about 5 or 6 years ago, my goodness, maybe a little bit more than that now, where I challenged the leadership of the 3 academies that environmental health cross the borders or the boundaries of basic sciences, medicine and health, and engineering, and that we needed to bring them together if we were ever going to be able to address some key environmental and public health issues. And it's actually turned into a very exciting effort that's ongoing here for that committee. Now, I really want to thank all of the members of the committee who helped organize this workshop planning committee. Especially, I have to call out not only and refer again to Rod Page and Wendy Shelton, who have worked tirelessly with me, as well as with Ruth Cooper, who is the Academy lead for this meeting with help from many of her colleagues. But I think it's been really quite a great experience to work together and as we developed this and came up with new ideas. And I have to say that I have a certain trepidation when I look at our program because it is so chock full And, after getting used a little to Zooming, where, you watch for an hour and then you get up and you dance around your room and then you go back and do something. If anybody feels like dancing around the back, there's plenty of room for that too. But anyway, so I'd like to give a little bit of background on the workshop. And it's going to really look at the potential role of companion animals as sentinels. And so we know that shared environmental exposures affect both people and their pets. And we think that that is really sorry guys. We really want to challenge the emerging field of exposure science, because we all know that it's not just one chemical at a time, and it's not just one point in your lives, and that we want to try to bring this together to try to accelerate the knowledge that we'll get related to cancer and aging. So topics for invited presentations and discussions include potential data sources to really understand whether our companion animals can serve as sentinels, state of the science for biomarkers of exposure and the use of biosensors, the best practices for collection, storage, and analysis of biosamples to assess exposures, different strategies for standardizing and sharing and aggregating health records and relevant metadata, and current policies and regulations related to monitoring and mitigating environmental exposures, and the role for prospective interventions based on companion animal data. So just a quick overview of the program for today. This is the workshop, and the welcome and the overview, but the first session, after we have some of our introductory presentations from Ned Sharpless, the director of the National Cancer Institute, is going to be about the history and current state of the science of environmental exposure effects on aging and cancer, and also methods and current studies. And first, there's a virtual poster session, and then there is an in-person poster session with a reception tonight that I hope everyone will participate in. And I urge those on the line virtually to participate as much as possible as well. Tomorrow, we are going to have look at the relevance of companion animal exposures to human cancer and aging. And again, a virtual poster session early and later in the day, an in-person poster session. And we'll also be talking about accelerating cross-species comparisons, opportunities, and challenges. And then on day 3, and I forgot to reiterate the point about using the discussion box below the webinar live stream to submit questions, or use the microphones in KEC 100. And again, since we are virtual as well as in person, please be sure to speak at a microphone so that the other folks can hear us. But on the last day, we have a really important session, I think, on the issues of equity, ethics, and policy, which I think will be instructive to all of us and may give us some new ideas as well. We'll then have facilitated discussions with session moderators, and this will really be an opportunity for people to get together in small groups in person, and we will have facilitated discussions on the web as well available. And then the last session where we'll identify research gaps and setting a research agenda with recommendations and next steps for the path forward. And then we'll have some closing remarks, which means at the very end, I'm going to try to pull it all together so we kind of know where we came from, where we are, and where we're going. So with no further ado, I'd like to introduce Dr. Ned Sharpless of the National Cancer Institute. I never even noticed that. I kept looking back there.
Thank you. Morning, everyone. Thank you, Linda. It's great to be here. How am I going to work this thing? Let's see. Doing something in person. How do you work the clicker? It's been so long. I think this is a great topic. The intersection of cancer and aging is something I've been interested in my entire career, and certainly the use of novel model organism systems is an important development. This is also an area that I think maybe it's important to say it's an interesting time in cancer research, right? The National Cancer Act in 1971, 50 years of basic understanding of cancer. And now I think if you really look at it, there's this real sense of rapid progress in cancer. So, the national mortality is declining at a rate that is increasing over the last few years. We've seen new advances in diseases like melanoma and lung cancer that were sort of refractory and intractable 10 years ago, and are now diseases where we cure a lot of patients, not just treat, but cure individuals. When I was at FDA, we saw, like a third of the business of the Food and Drug Administration in terms of new drugs and devices are cancer-related for oncologic indications. And it's a very, very, very exciting field. And so I think we'll come back to look at this period right now, the decade that preceded now and maybe the decade to come as a real sort of golden age of cancer research where we saw a rapid, promulgation of new therapies and new ways to prevent, diagnose, treat cancer. But, one part of that, so I think in general, there's an exciting trend in cancer research, and certainly having a president now who said he would like to end cancer as we know it as a top domestic priority of this administration is very exciting and galvanizing for the field. But if you look within the whole giant sphere of cancer research, there are some pockets that are concerning. So, for example, we still have significant health disparities in cancer outcomes. We have some patients that do quite well with a certain diagnosis, and other patients do poorly for reasons that have more to do with wealth and access and race and ethnicity and rurality than they do to, what's the ideal treatment for that tumor. So, cancer health disparities is a pressing issue for our country. But this topic of environmental carcinogens, I think, is also an area where the NCI continues to be quite worried, and where I've talked with my colleague, Rick Wojcik, who runs NIHS, and we share many of the same concerns, Linda's successor. And, one thing that's changed my thinking in this field is having spent time at the FDA. Prior to going to FDA, where I was acting commissioner for 7 months during 2019, carcinogens were kind of a biological research problem. It was like, how does agent X cause cancer? Why? That's an interesting scientific question. But then when you go and sit at the food center or the animal center at FDA, and you daily see how they're tasked to make regulatory decisions, what level of PFAS should be in the Cheerios, questions like that. That every day the FDA has to struggle with such issues. And then when you interact with the EPA and see how they have to similarly deal with such issues, you realize the pressing importance of this topic and how it has a wide impact on American life and is of great interest to both scientists and regulatory decision makers in this field. And so I think that experience has significantly evolved my thinking in this area and led me to believe, to think that the scientific groups like the NCI and NIHS, could we do more to support the scientific underpinning of this topic to help inform these policy decisions? So one of the things I asked our groups to do is sort of look at what we support already, both extramurally and intramurally at the NCI, and this is a very brief summary of that. So we surveyed about our extramural divisions, the Division of Cancer Biology, the Division of Cancer Control and Population Sciences, Division of Cancer Prevention, et cetera. And identified about 80 grants that are targeted on environmental carcinogens at present. And here's sort of a summary of what they look at. These things like microbiome and gene-environment interactions, using various omic approaches. And I think it is many of them using rodent animals as a model. I think it is a solid extramural portfolio. But is it the right size? I think that is a fair question to ask at any given time. Additionally, we have a significant amount of both extramural and intramural larger studies, sort of top-down driven science, some of which are listed here. So examples are the NCI Cohort Consortium approach, which is a collection of, founded in 2001 of these many large cohorts, many of which have an environmental agent focus. We have large scale, and these are all cohorts that are sort of more than 10,000 participants or greater that are cancer focused. We have the IARC Monographs program that we've supported that is since 1982, that is an effort to try and classify environmental carcinogens that I think many of you are aware of. The Connect project, which is recently started, is a large effort to monitor on the order of 200,000 individuals through time with the collection of biospecimens and also collection of detailed questionnaire information about environmental exposures. We have the Ag Health Study, which is, I think, really been a landmark study in terms of the environmental risk of pesticides and cancer, for example, and many, many more. And then on the left here are a few examples of intramural research from DCEG, our Division of Cancer Epidemiology and Genetics, And there are things like, showing smoky coal causes an increased risk of lung cancer in non-smokers and risk of arsenic and bladder cancer, for example. So a lot of great work there as well, for which the NCI, I think, is well known. Additionally, we have an interest, I think, of importance to this group in companion animal models. So this is work that has been developing for some time now at the National Cancer Institute. I think, like the National Institute of Aging, the NCI has wondered if companion animals, particularly for the NCI canines, could serve as useful models to help understand many aspects of cancer. I have to tell you, one of the things we talk about most in this project is the ability to do immuno-oncology trials in dogs, because immuno-oncology trials are very hard to do preclinical testing for. The rodent models are not generally great for such agents, and so often they go straight to human clinical testing, and that is inefficient. At one time, we probably had 1,000 or more different PD-1 plus Agent X trials going on in the United States, which did not make sense without better sort of preclinical credentialing. So there's a hope that some of this could be rescued with dog trials, and the NCI is invested in the creation of sort of clinical trials networks for dogs, and also to even developing certain agents like a dog-specific, a canine-specific, antibodies for immuno-oncology use. But additionally, I think it's not lost on this group that the sequencing of dog tumors can be quite informative, and as mentioned, companion animals share the environment of their hosts, and that therefore could be informative in terms of environmental agent research, and we believe that's very important. This has many parts to it, but perhaps one of the most important that I would highlight today is that we have now created a large integrated canine data commons. It is part of the Cancer Research Data Commons, our gigantic data commons portal. It lives in the cloud, and that now keeps both clinical data on dog trials as well as dog sequencing information, and can be contributed to the same way, the Cancer Research Data Commons has the 1,000 genomes or whatever. So that, I think, is a resource that will continue to grow, and I hope will be of use to this community. Back to this topic of environmental agents, I believe, as said, that this has worried me for some time, and we recently convened a meeting to sort of hear the state of the science, co-hosted with NIHS, on environmental agents and breast cancer. And it was a very lively sort of day-long meeting that had many external speakers, and the proceedings of that are published at cancer.gov for those of you who are interested. But I think it was informative in some ways. I think we heard a lot about scientific approaches that have existed for a while, large rodent studies, for example. And we heard a lot about potential new ideas, new approaches, but some of which have not yet come to fruition but are very promising. But I get a statement that I think probably 2/3 of the speakers said at that meeting, both intramural and extramural and from outside the NIH, was we sort of need new thinking. There's a sense that we need new ideas and new approaches and new ways of trying to link environmental agents to a given cancer. And I fully agree with that. I think that the time-honored troika of biological plausibility, rodent animal research, and cohort study has been useful for assigning carcinogens to cancers, but it is not perfect, and it could be improved. And so that was a lot of discussion about where to next and how can we get better at this in a way that's efficient and cost-effective. And so I think companion animals as a model organism is one approach that we'll talk about a lot. Here are 3 other things that I think are interesting that we talked a lot about at this meeting in terms of the emerging science of mutational signatures, the ability to ascribe a carcinogenic insult to a specific cancer based on a pattern of DNA mutation, somatic DNA mutation found in the tumor. There's been a very exciting set of studies, for example, linking aristolochic acid to renal cancer in Eastern Europe. Recently, the NCI had an intramural paper showing a lack of a tobacco signature in people with non-smoking lung cancer, suggesting that they're not getting their tumors from secondhand smoke, or at least that's a less important cause than perhaps some other causes in that population. Ultra-sensitive detection, I think this is the power of very sensitive instruments to find 1 part per trillion or 1 part per 100 trillion of certain environmental contaminants is both a blessing and a curse for this field, because this level of sensitivity is unprecedented, and how to use that information is still unclear. The power to really do data aggregation, to use trials like All of Us and the Connect study that are very large and have long-term follow-up, I think can fully empower, even if the trial wasn't specifically designed for carcinogen research, I think can still be highly valuable. And so these are, and the topic of exposed zone monitoring, by the way, that was something else that we talked a lot about. But I left that meeting with the sense that that this is sort of messy science, that there's a need for further thinking in this area. And I think many of us shared the view that new approaches are warranted. And I frankly believe this is an area where the NCI could use external perspective. So meetings like this are very, very important. But I think we will, in the future, likely have to convene a group of august thinkers to help us work through some of these issues. And in a sense, sort of come up with a new Koch's postulates, if you will, of assigning a carcinogen to a cancer that's molecularly precise and modern and incorporates the time-honored troika I mentioned with sort of these modern new technologies. I also want to before I finish, plug this meeting. Tomorrow and Friday, the NCI has a meeting on mutational signatures that is open to the public, and I think 700 people or something have signed up already. And it really delves into this topic of how you can identify the carcinogen that caused the cancer based on the somatic mutational signature of the cancer. It's very exciting science. It's very complicated biology. It involves a lot of math, for those of you who like linear algebra, time to dust off the textbook. But it's fascinating research, and I think is a very powerful tool that is already being used in the science field, and I very soon predict it will start making its way into regulatory decision-making in other areas that are important. So for those who are interested, please check that out. With that, I will wind up, and I think we have time for questions, Linda, right? Okay.
And when people are up here, they don't need their questions. We are 6 feet. So do we have any questions? Myrtle? Please go to mine. There's one right next to you.
So, good morning. I'm wondering about the investment that the NCI is making, or is it continuing the investment in the molecular characterization of canine tumors, in that one of the advantages we'll have is understanding molecular origin and how that may pertain to cancer types that really aren't present in dogs and companion animals, but yet have very similar molecular footprints and maybe even some similarities with regard to metastases. So I was just wondering about that investment and how you envision continuing that investment and what the strategy might be.
Yeah, no, I think this is really important. I almost believe that for the study of a cancer, whether it be of a dog or a human or any model organism, characterization of the somatic genome of the tumor is sort of the state of the art. I mean, you have to do that without doing that now. That's also, I think, basically true now in clinical medicine, that to take care of a patient with advanced cancer who has no obvious standard of care, molecular characterization is, should be a must, because it's just so critical to understanding the biology. Two tumors that look the same under the microscope may be very, very different depending on the driver events that cause that cancer. But the good news is, 10 years ago for this topic, we needed a gigantic, moonshot kind of cancer genome atlas project because sequencing was very hard and the data analysis in particular was very hard. And that is no longer true. Now sequencing is a commodity. It's widely available. It's done well by many, many both commercial and academic labs. And the data analysis is still challenging, but there is enough expertise that, largely through NCI investment, that those individuals are widely available for scientific collaboration. And so, one of our hopes is that we've built the genomic data commons for canine tumors. We would now like individuals to upload data to that to make that resource more and more useful. And there already are a number of genomes there, but we hope the somatic genome number will grow very quickly the way it has for human genomes in the Cancer Genome Atlas.
Hi. Yeah, so I just had a quick question with
Excuse me, could you please introduce yourself?
Oh, yes, sir. James DeGregori from the University of Colorado Anschutz Medical Campus. So, we're really good at detecting mutagens and their signatures, but what about selectogens? In other words, there was even a recent study, and my apologies to the author because I don't remember their names, but they showed that they screened through a large number of carcinogens-induced mouse tumors, and only a small fraction of them actually changed mutational signatures or mutational burden, so suggesting that many of them were acting more by altering context than really altering the mutational landscape. But we don't seem to have really good methods to detect those.
Yeah, I think that's a terrifically interesting science. Is that, by the way, I should mention one of the president's interests here in cancer research is greater cooperation with the United Kingdom. So the U.S. and U.K. have pledged to joint interactions around cancer based on an announcement that Boris Johnson and President Biden made at the G7. And so one of those things started with a scientific summit that happened about a week and a half ago. And one of the sort of 6 topics we picked was this topic of, it's very interesting to our UK colleagues as well, of environmental carcinogens. And that effort was in part chaired by Michael Stratton, who is famous for, promulgating this idea of mutational signatures along with colleagues at the NIHS in the United States and the NCI. And this was a lively discussion, is what about these things that clearly cause cancer? They're clearly carcinogens. They're environmental agents that clearly induce cancer by any reasonable sense of that definition of that word, but they're not mutagens. They don't damage DNA. They don't break DNA. They don't bind to DNA. They don't have an ascertainable signature. What are those things? And, they need a name. Selectogen may be as good as anything but they need a scientific approach. And I think that is certainly ongoing, interesting science where I believe some, both model organism research and human studies will be very, very important. I predict there'll be a lot of these different kinds of molecules, and they will have different mechanistic impacts. So there won't be sort of one way of being But I think a good example, for example, is agents that sort of seem to cause chronic inflammation, and that leads to a variety of cancers. But there's no clear damaging signal. But I predict there'll be many other classes beyond that.
Do we have any questions from the web? Ruth?
Yes, Lori.
Okay. Thank you, Lori.
Sure. The question is from Roy Jensen, and he says, what do you think of developing a cancer center program for vet schools to facilitate developing companion animals as cancer models?
Good to see Roy virtually, our old friend. I think certainly we have been interested in supporting extramural networks. Whether they should be cancer center based or extramurally based is something I think we'd have to think about. Certainly some of the great companion animal cancer research programs nationally in the United States are affiliated with cancer centers. So I actually gave virtual grand rounds at UC Davis yesterday, which has incorporated its vet school kind of into their cancer center program in a very formal way. But not all of them are. There's some terrific vet schools that are not linked to a cancer center, and I think we would want to be able to include them in this research as well. So, the best administrative framework to lead to the most robust science is a topic for discussion. But I do think it is an area where continued support and even maybe increasing support from the NCI extramurally is valuable.
I'm going to ask one question, take the purview of the chair. So this issue of selectogens, I mean, in experimental experimental bioassays, one of the major approaches that's being suggested now is the use of the development of key characteristics. And if you find there have been about 10 defined for carcinogenesis, and if in fact a compound has any, or let's say several of those key characteristics, it is likely to be a carcinogen. Is that something that you're interested in?
Well, I think simpler in vitro assays to identify carcinogenic compounds would be welcome, but that often requires, I think, a biological understanding of how these things work that we don't fully have. I think things that damage DNA, that we got, right? There are a number of in vitro assays that work really well and have now become, sort of the industry standard for developing of new therapeutics or in, say, food testing, for example. But for these emerging classes of carcinogens that we fully understand, I think the performance of an in vitro bioassay could be useful, but we sort of have a little work to do to match up how well they predict, cancer risk in people.
Well, I wasn't going really directly to new approach methodologies and all the in vitro. There's lots of different in vivo studies where, in fact, there are opportunities to potentially classify new compounds or new mixtures of compounds, which may be even more important, with information whether, for example, they bind to specific nuclear receptors, which is one pathway that seems to be very important, or whether they generate oxidative stress, those kinds of things. So
Yep.
be an interesting thing, I think, to talk about more.
I agree.
So I'm curious how you think of NCI maybe
Please introduce yourself first.
sorry.
Matt.
I'm Adam Boyko. I'm at Cornell University and with Embark Veterinary. And I was just curious what your thoughts were about how NCI could work with private companies that maybe are doing some cutting edge research and data. Like in this space, you've got Phytocure and Vidium that are profiling lots of canine tumors and amassing a lot of data that way, as well as working with veterinarians to bring that to the clinic.
Yeah, so there are a number of ways we can work with industry, and we have a very, it's probably true the NCI works more with industry than just about any other biomedical research entity in the federal government. I mean, we have extensive interactions in terms of both a CRADA mechanism where we can, do in-kind support for trials. We have data contribution efforts. We have a very large SBIR program to support particularly early stage companies in development. So I think there would be a number of approaches. Of course, NCI interactions with industry have to be managed at arm's length in an appropriate way, which is very manageable. But where we've had some success are, if a company has a novel therapeutic or device, we can work with them to help develop that. If a company has large sets of data that they would like the public to have access to, we can be a portal for that. So, for example, Foundation Medicine, the somatic genome sequencing company, has given us on the order of 17,000 genomes that are now available through the Genomic Data Commons that are available to researchers because they believe that's good for the biomedical research community. We agree. So we can help the development of compounds. We can help with holding data or making data available. And lastly, we have funding mechanisms to support particularly early stage companies through the SBIR program.
Hi, I'm Rod Page from Colorado State University. Dr. Sharpless, you mentioned some need for infrastructure for cohort studies. I don't know if you have specific ideas in mind for that or if that's a part of a larger conversation, but I just would support that and wonder if there's a way to consider how NCI might continue to build their already robust program in companion animal cancer with cohort study support.
You're speaking about therapeutic studies or about sort of observational cohort studies?
Observational cohort studies.
Let me take that back. As I said, I think a lot of our focus initially had been on therapeutic, networks, for example. But we do have this data commons. We do have this interest in collecting data, and I think there might be a way to make that more useful by also collecting some modest amount of demographic data, if you will, on the companion animals that matches the somatic genomes. The typical approach to that is to sort of standardize it across multiple centers, is to think about the number of questions, the standard set of questions you'll ask, and standard way of collecting genomic information, and that might be a good idea. We will have to think that over. Thank you, Rod.
Well, I'd like to thank you, Ned, very much for participating in this, and hope you can stay around a little more today and answer questions.